cobalamide, it needs it to convert it into the tetrafolate to repair.
Very nice. Thank you. What else does the folate do? Or a screen, something on the screen. Why do I bring up folate now? Yeah.
So folate is vitamin B9. It combines with vitamin B12 to form S-adenosylmethionine, which is a universal methyl group protein.
That's it. That's the answer I was looking for. Thank you so much. S-adenosylmethionine makes your life who you are. Most of it. Some of it. It's a great little small molecule. SAM. S-adenosylmethionine. Right? Okay? Because we're going to study histone methylation, so I will bring this up. Now, methylation. We talked about yesterday. Which methylation? DNA methylation. Yesterday we talked about DNA methylation. Today we're talking about
Histone methylation.
what is common in both? Methylation is the key to many things. I told you guys yesterday, we are animals. Our planet is a carbon-based planet, right? It's a carbon-based planet for the most part, isn't it? Some people out there, if it gets this is getting recorded, right? It's on YouTube, you are saying wrong things. Maybe I am, but it is a carbon-based planet, right? Methionine is a great thing because it does methylate most of the time. Sorry, and you will see in a second why methylation is so critical. Okay, before I go forward, sorry, I want you guys to think a little bit. We talked about COVID virus, right? You guys know anything about COVID viruses? How did humans evolve? How many of you believe that there was an Adam and Eve suddenly came to this planet? No. Nobody believes. How many of you think suddenly Ram appeared? Many Hindus here. Right? What do you guys believe in? How did humans evolve? Sorry, I shouldn't be speaking this stuff, but it's just sometimes I have to. In Texas, they don't believe in evolution. They don't teach evolution. In Texas, United States, a very rich state that produces a lot of oil, that makes a lot of new things happen in this world. They don't believe in evolution. Can you believe it? I was taken aback when I heard that. I really because Republicans think that God created Adam and Eve one day, right? Once, sorry, but that's true. Some people believe that that's true. Maybe it's true. Maybe I don't know. But if you believe in evolution, and I for one I do, right? What happened during evolution? How did you evolve? Okay, I think I'm asking a far-fetched question, so I don't think I know the answer to this. But you know, in our genomes, I'm talking about viruses, there are lots of viral elements. Do you guys know this? How many of you know this? You knew this. Okay, what are the viral elements?
Some structural proteins and non-structural proteins are there. And the genetic material like positive signs and negative signs are there.
Okay.
So some non-structural proteins play a role in the resilience of that virus to attend the host cell.
At least you tried. Thank you. That's not true entirely. There are lots of our genome is actually made of something called repeat elements. You guys heard of this? Most of those repeat elements come from viruses. So we, as part of evolution, humans actually did what? We took viral DNA in our genomes, right? So there are a lot of repeat elements in our genomes of 3.3 billion and 6.6 billion bases that came from viruses. These are called retroviral elements. Yeah? Good. But why are they not are they expressed? They're not expressed normally, because if they did, then we would not be who we are. But they're there. And in fact, they play a huge role in epigenetic regulation of the gene. But normally they're kept silent. They're kept silent. Why am I telling you all of this now? I was going to talk about methylation. What did I say yesterday about DNA methylation? Do you remember? What does a DNA methylation do to gene? Where DNA methylation is, what does it do? Huh? You guys don't know, right? We'll talk about this tomorrow. The DNA methylation silences genes, right? I mentioned that earlier. And part of histone methylation, as I mentioned earlier, also silences genes. And how does the methylation happen? By S-adenosine methionine, that's a key substrate for this, right? A key cofactor for this. Enzymes that are histone methyltransferases. So that's why they're so critical. They're so critical because without methylation, we would actually not be who we are. Okay? Did that make sense? Okay, alright. Alright, let's go to histone methylation because this is going to become a little didactic for now. So, we saw histone tails, we saw they get methylated at different positions. Methylation happens on arginines and lysines. We saw this thing earlier. Where does methylation take place? It's in arginine and arginine residues of the histone tails. Which molecules help in methylation? That's anybody's guess. Methyltransferases. S-adenosine methionine is the donor group of the methyl. That's what gives you methyl for the enzyme to put on wherever you want to put. That's why vitamin B12 is so cool. You need vitamin B12 because you need to make these things, right? This is part of your daily life. Okay? That's why you need to eat vegetables, guys, so you can get vitamins of different kinds. Vitamin B12 is critical. Okay? What's the consequence? It's a context-dependent transcriptional activation or repression. Methylation, I said earlier, will be monomethylation. But methylation happens on lysines or arginines, and it can be mono, di, or tri. Right? Are there chemists in the group here? Huh? And H3 is? What is an H3? Ammonia. Why H3? Nitrogen has 3 valencies, right? Open. Chemist, come on. This is, huh? Sorry, what was it? 3 copies of the that's right. Isn't chemistry amazing? Makes you have 3 different methyl groups. But in the genome, you can find monomethylation, dimethylation, or trimethylation. How does that happen? If the valence is open, why doesn't it go all the way to trimethylation? Right? Good question. Please go look at the answer of that if you can find it. So, interestingly enough, these histone tails will have, when you look at it, when you find it, you'll see only monomethylation happening at some points, and some sites it will have the same residue you'll have the trimethylation. When you have H3K4 monomethylation, it goes on enhancers. H3K4 monomethylation is a marker for enhancers. What did I tell you about enhancers? Regulate gene is the most important regulatory element, at least in my mind. It makes your eye, it makes your liver, it makes your intestines, and to identify enhancers, you look at histone H3K4 monomethylation. Okay? By a method called ChIP-seq. I was supposed to teach you guys methods too. That's part of the tutorial, but I don't think we'll have time now. Tomorrow maybe we'll do it. So who writes the mark? Histone methyltransferase. Who raises the mark? Histone demethylases. There are specific demethylases for tri, di, or mono. I did make it very complicated for you, didn't I? Then what reads the mark? There are domains called chromodomain, just like bromodomain, chromodomain, bromodomain, chromodomain. There's enzyme ZD1A. That's an enzyme which is a very well-known histone methyltransferase. It recruits a complex of proteins. It's called ZD1A complex. Okay, fine. There are multiple such complexes, by the way. ZD1A, MLL1, MLL2, MLL3, MLL4, MLL5. Excuse me. Okay. K4 trimethyl is recognized by a domain called PhD. There are lots of different domains that recognize methylation, by the way. So PhDs are the major ones, and chromodomains are other ones. So H3K4 trimethyl is recognized by the chromodomain. That helps recruit all of these beautiful things, which is TAF3. TAF3 is would be Tata associate factor, one of them, right? So in the original one, so what I told you, in some of those chromatin remodeling complexes, there was bromodomain proteins, and these is chromatin. Why does this matter? Why is Mr. Arpan showing us this? You know why is he showing you that? Because a Tata binding protein gets recruited where K4 trimethylation is marked. Every single gene that is going to be transcribing, they're going to get activated as a K4 trimethyl mark. How cool is that? Right? I'm flagging every gene that I want to be marked. And then that will help me recruit the Tata binding protein and the Tata polymerase and da da da. How cool is that? Right? I will need an acetylation to move a nucleosome, but I will need a K4 trimethylation to recruit my Tata binding protein. Yeah? Yes? Yes? No? Is it cool? Is it not cool? Just because Kunal saying it's cool, it's cool. So every single gene, actually, this is used as a marker for gene that is can be activated. If I want to find in by your cell what gene can be activated, I will look for H3K4 trimethylation. Okay? I will look in a genome where H3K4 trimethyl is marked. And I'll tell you the method tomorrow by how we look at it. But this is what you do. You map this. You map H3K4 trimethyl marked nucleosome to say, where is my nucleosome that's got H3K4 trimethyl mark? And I'll, boom, that is going to be transcribing. I can tell you by confidence. That's called epigenome. That's the whole crux of this approach of this course. We learn what his epigenome marking is, so I can tell what's going on where in my genome of 6.6 billion bases. Did you get it? That's one of the things at least. Okay? Is that clear, by the way? Is that not clear? You got this? I want to show you guys an experiment. Okay. Let me see if I have that experiment here. I may not have that experiment. I'll bring it tomorrow though. Let's see if I got this. No, I didn't put it on. I want to show you a classic experiment that is done at Rockefeller University to demonstrate that H3K4 trimethylation will actually activate the mark. Is this here? No, this is not it. I wish I did. Okay, I'll show this tomorrow. But what do you guys, what did you guys get my point? Okay. All right, we'll do this other lecture tomorrow. I'm running one lecture behind. Where were we? Okay, we were here, right? So K4 trimethyl is critical for activating of gene transcription. H3K, similarly acetylation is critical for recruiting the nucleosome remodeling. If I don't have both of them, I'm not going to be able to do my gene transcription, right? I need acetylation to recruit my nucleosome remodeling to move the nucleosome. I need my K4 trimethylation for it to sit there and active and recruit Tata binding protein. Is that cool? That's called histone code. I need both of them. So both of them is a very simple answer, but I can have many of these combinations of the marks that are needed at specific nucleosomes to do specific things. In this particular case, I'm just telling you an example. You need an acetylation to recruit. What did you need the acetylation to recruit? We just spent like 15 minutes before this doing that. Huh? Nucleosome remodeling complex, that's right. That had a bromodomain protein that reads the nucleosome acetylation, right? Histone acetylation. And why do I need the H3K4 trimethylation? Tata binding. If I don't have both of them, I'm not going to be able to activate it, right? So K4 trimethyl alone cannot do it. K4 acetylation cannot alone do it. So I need both of them, right? I need an acetylation and amortylation together to do this. That's why we are built in this way. That's why nucleosome has tails that get modified in many different ways. How cool is that? If I have a K4 trimethylation, I cannot have a K9 trimethylation. They're never found together in the same nucleosome. That's also a code. Non-existence of one is absolutely crucial for the existence of the other one. All right? How cool is that? That's why David Ellis was brilliant. Okay, we studied this. Da 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 da. Then I need a demethylase to remove the mark. Okay, if K4 methylation is there, I can't have K9 methylation, I told you, right? So now K9 methylation, when it's put on, it's recognized a protein called TRIM28 that repulses the SATDB1. That SATDB1, K9 trimethylation. SATDB1 is an enzyme that does K9 trimethylation. Okay, you don't have to remember the name, but just make it easy for yourself. It recruits. And then it is read by HP1, which is a chromodomain protein that specifically reads K9 trimethylation. You had a PhD domain reading a K4 trimethylation, you had an HP1 that's reading K9 trimethylation. This is exactly how retroviral elements keep silent in our genomes. The cell puts a lot of H3K9 trimethylation mark on all the repeat elements in the genome. Okay? Is that cool? So K9 trimethylation, when it's put on, then it recruits DNA methyl transferase as well. And also DNA methylation is put on. That's another silent mark. So K9 trimethylation along with DNA methylation together will make a gene repress for a long, long time. So it's a permanent silencing of retroviral elements is done by K9 trimethylation and DNA methylation. Now you got it? I talked about transient activation. I talked about permanent silencing. There is no permanent activation. Okay? Why? Because we have the desire. We need it. We need to silence the retroviral elements. Otherwise, they'll keep hopping around the genomes and they will create a lot of genomic instability. They'll create DNA damage. Then we will have cancer and we will all die. So in cancers, a lot of times, these retroviral elements actually get activated because there is no more DNA methylation. Doom, damn, doom. You're dead. That's how mutations are caused. That's one of the mechanisms of how mutations happen in cancers. Isn't that cool? Retroviral elements get activated. Okay. In this particular case, there is no protein made. Only you make in this cases when the gene retroviral elements are activated, it makes only an RNA. This is non-coding RNA. And those non-coding RNAs activate a signaling pathway called cGAS-STING cell signaling pathway that activates the immune response, that kills the immune response of the cell, meaning the immune cells cannot activate a cancer cell and they cannot kill it, so the cancer cells survive and they go on to do your cancers. So, just told you an example of something I was telling yesterday, that you don't need proteins all the time. You can actually activate the phenotype by just putting a RNA. Isn't that cool? I told you like a lot of things in last 1 minute just now. These are key things, how things work, how cells work. Okay? Right, cool? So that's why epigenetics is so cool, guys. K9 trimethylation. You know what? HP1 will recruit an enzyme called SUV39H1, and that will mark the next nucleosome and the next nucleosome and the next nucleosome and the next nucleosome. Remember, heterochromatin has all the packed things next to each other. That's for that reason. So all that heterochromatin has nucleosome after nucleosome, methylated, 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 methylated. So a long stretch of several MB genomes in your genomes are silent because of, okay? Is that cool? My daughter says cool. So I sometimes say cool. Okay, so the moral of the story is, what's on the slide? Can you guys read the slide? You guys got that? I told you about gene repression. I told you about how heterochromatin works. And I told you about how activation happens through. Isn't that cool? Epigenetic regulation. This is crux of epigenetic regulation. Today's class is how epigenetic regulation actually occurs in your cell all the time, in every single cell in your body. How many cells in your body? Who was listening to me this morning? 37 trillion human cells. Every cell has this damn epigenetic regulation going on in them. Isn't that cool? It's going on in a specific way. Your eyes are firing different things, your skins are firing different things, and your intestines are firing different things. I love it, right? It's beautiful. This is why epigenetics is so important. Okay? All right, the moral of the story is we got H3K9 methylation that's silent, H3K4 methylation that's active. You have different readers that do different things, different molecular outcomes in the phenotype, and that goes back to all the things you've learned earlier. I'm not going to go into this because I'll send you the slides. You guys can read this. This is a summary. You can have this. We'll come back to this tomorrow. I don't think I want to bother you anymore. Good job. Thank you. 4 o'clock. Exactly. I want any questions? Any questions? Actually, I want to leave you with something else. I'm sorry. I'll take 2 more minutes. I'll start with that tomorrow again. We'll end with a YouTube video because we are a generation of YouTubers here. How many of you dream to be influencers? None of you? Okay, I want to show you this before you leave. Just because you can dream of this today. Ah, man, come on, you gotta move. No? It doesn't move? The YouTube generation has to be shown a YouTube video before they go home. Is it because the internet's not working? Sorry, what's wrong with this? Huh? I can search directly? कहाँ पे? Oh, sorry. I'm blind. No. I don't know if I can find this video. Okay, let's see. Happy logos. What is that? So my dream is not going to come true today, I guess. No. Let's go. Epi logos. Yeah, you can see something, right? What do you see on screen? This is your genome, guys. We started with it. I was showing you IgV, right? I was showing you just genes. I want to end with showing you epigenetic elements on this. Do you see this? What do you see? You see a blue tower, then you see lots of grays, and you see green, you see red, you see yellows, you see greens. This is your epigenome. Is that cool? I'll belabor upon that tomorrow morning again. But I find this extremely cool. Why do I find this extremely cool? No, nobody knows this, right? Look at these genes. There are genes going. I'm going to close this up. Okay, right here. See this right side? There is a gray region, right? Gray means silent. H3K9 trimethylation or K27 trimethylation. Red peaks are essentially histones that are H3K4 methylated. You see, it happens right at the start site of this gene. You see this, where my arrow is? That's the start site of gene called PARK7. You see the red building right there, right? That's where the gene transcription is going to start. I just told you. See how cool that is? Because that's just separated from the left gray right there. So the cell knows exactly where I need to stop and where I need to start. The yellows are the enhancers. And the greens are telling me that region is being transcribed by RNA polymerase. Green is a marker for RNA polymerase transcription. So this gene has an active transcription. It has an active mark here. It has another active mark right here. So some cells have one transcript being transcribed, the other one has another one. They have green here, they're making transcription. There are enhancers here, and this rest of it is silent. So the TNFRSF9 gene is actually silent in this cell. It's not making gene transcription, but the right one is. So the cell knows exactly where to start the gene transcription, where to stop it. It knows which gene to mark for activation, which gene to mark for silencing. You get it? And you can go through the whole genome and you see this marking is there. My lab builds these things all the time. We build these maps all the time. This is a machine learning based approach that makes you these maps. Okay, well, let's discuss tomorrow how you do this and how the method to do this. But I wanted you to appreciate that you have a gene linear and now you have an epigenome on it. This is what the whole course is about, to teach you epigenome. Okay, that's epigenome. Cool. All right, go enjoy, have fun. Thank you. We'll see you tomorrow.